Dear students, good morning to one and all. Today we are going to learn about define cell biology and cell theory and one of the cell organelle endoplasmic reticulum. Cell theory is the study of the structural and functional of the cell. It also known as cytology. Schwann is generally called the father of cell biology because he described the morphology and physiology of cell of the first time. Next we are going to learn cell. Cell is the basic structural and functional unit of all the living substances. The cell was first discovered by Robert Hooke in 1665 in Cork. The cell is living unit and life actually resides in it. The cell theory concept proposed by Sweden and Schwann in the year 1839. Cell theory states that all organisms are assembled out of the cell. Cell are organisms. They arise from pre-existing cells. The activates of the organisms are the outcome of individual cells. The cells are divided into two different types. One is in prokaryotic cells and another one is in eukaryotic cells. Prokaryotic cells, they do not contain defined nucleus. Examples, bacteria and some blue-green algae. Eukaryotic means a eukaryotic cell contains a defined nucleus. The chromatin bodies are enclosed by a nuclear membranes. So next we are going to learn endoplasmic reticulum. Endoplasmic reticulum is a network of membrane-bounded cavities, vesicles and tubules are distributed throughout the cytoplasm. It is concerned with the biosynthesis of protein and the lipids. It is a cytoskeleton of the cell. It is a cytoplasmic vesicular system. It is a transportation system of the cell. It is the function as the packing system. The term ecosystem was first proposed by Porter in year 1952 and Fullman. In the year 1945, the lake-like membrane of the endoplasmic reticulum was first seen in cytoplasm of chick embryo in cell. These are single membrane cell organelles. Endoplasmic reticulum considered as one of the component of cytoskeleton along with the microtubules, microfilaments and intermediate filaments. Location. Most of the endoplasmic reticulums are present in all the eukaryotic cells. These are found to be absent in mature erythrocytes, egg cells, red blood cells and bacteria cells. Next we are going to learn structure of the ecosystem. Endoplasmic reticulum consists of three main components. They are cristae, vesicles and tubules. The cristae means these are long, flattened, unbranched, sac-like structure. They are arranged in a parallel bundles. Their diameter is 40 to 50 millimicrons. They have ribosomes on their surface. They are normally found in a secretory cells. Next we are going to learn vesicles. These are rounded or oval in structure, having the diameter of 25 to 500 millimicrons. They are found in pancreatic cells. They are found in end of cisternae and tubules. Many vesicles are left free from the cytoplasms. Vesicles are surrounded a coating protein called COPI and COPI. Next we are going to learn tubules. These are smooth-walled and highly branched tubular space having a diverse forms. They have a diameter of 500 to 100 millimicron. They normally occurring in non-secretory cells like muscle cells. The membrane of endoplasmic reticulum is made up of a two-comp layer. One is a lipids, another one is a protein. It has a fluid mosaic structure. It is 50 to 60 Armstrong thickness. The membrane of endoplasmic reticulum consists more than half of the total membrane of an animal cells. The membrane of endoplasmic reticulum is continuous with a plasma membrane, Golgi apparatus, and a nuclear membrane. Endoplasmic reticulum is classified into two types. They are granular or rough endoplasmic reticulum and the agranular or smooth endoplasmic reticulum. So first we are going to learn rough endoplasmic reticulum or granular endoplasmic reticulum. In some endoplasmic reticulum, a spherical granular-like structure called ribosome are attached on their surface. It mainly consists of cisternae. The membrane of rough endoplasmic reticulum forms a large double membrane sheet. This type of endoplasmic reticulum is also called granular endoplasmic reticulum. The binding sites of ribosome on the rough endoplasmic reticulum is called translocon. It occurs in almost all cells which are actively engaged with protein synthesis, such as liver cells, pancreas cells, and plasma cells. It is a form of flattened sex. So next we are going to learn smooth endoplasmic reticulum or agranular endoplasmic reticulum. Ribosomes are not attached with a membrane of this type of endoplasmic reticulum, so the surface of the endoplasmic reticulum is smooth. It occurs especially in those cells which are almost inactive in protein synthesis. It well developed in cells that synthesize steroid hormones at a system of tubules. Next we are going to learn function of the endoplasmic reticulum. Endoplasmic reticulum perform the following functions. The first one is a mechanical support. The endoplasmic reticulum divided the fluid content of the cell into different compartments by which is give the mechanical support of the cells. Hence it is known as a cytoskeleton of the cells. The next one is a transport. Endoplasmic reticulum act as a kind of circulatory systems involved in the import, export, and intercellular circulations of various substances. By this process, protein, lipids, enzymes, etc. The endoplasmic reticulum functions as a cellular circulatory systems. The third one is a protein synthesis. Ribosomes are protein factors. Amino acids are assembled on ribosomes to produce polypeptide chains. The next one is a formation of microbodies. Microbodies are small granule bodies filled with an electron dense granule rich in peroxides, it including peroxisomes and glyoxisomes. They are formed in a protozoa, yeast, liver, kidney, and higher plants. The next one is the synthesis of cholesterol and steroid hormones. Endoplasmic reticulum is the major site for the protein synthesized cholesterol and steroid hormones. In the testes, ovary, and adrenal cortex, the smooth endoplasmic reticulum plays the major role in synthesis of steroid hormones. The next one is the lipid synthesized. Endoplasmic reticulum synthesized triglyceraldehyde and phospholipids. It also stored the lipids. The next one is detoxifications. So detoxifications refer to the reduction of toxic properties of chemical such as drug and pollutants. So detoxifications occurs in endoplasmic reticulum of liver cells. A metabolic waste products such as fatty acids, steroids, and also detoxified by the smooth endoplasmic reticulum. So next we are going to learn differentiate the smooth and the rough endoplasmic reticulum. Smooth endoplasmic reticulum, the surface smooth endoplasmic reticulum not stunned with your ribosomes is called smooth endoplasmic reticulum. The surface of smooth endoplasmic reticulum is smooth. It's otherwise called a granule. It occurs in inactivity of cells. It's more tubular than rough endoplasmic reticulum. The membrane has a fluid mosaic structure. The next we are going to learn rough endoplasmic reticulum. It's otherwise called a granular endoplasmic reticulum. It contains 30 million ribosomes in liver cells. Rough endoplasmic reticulum basophilic. It is made up of flatted sacs. It consists of membrane, lumen, and ribosomes. The membrane has a fluid mosaic structure. It has more concentrated near the nucleus. It directly connected with your nuclear membranes. So, thank you, students.